Hello, everyone. Welcome to RegenMedGlobal, the podcast, where we bring you the latest and greatest in the field of medicine and research. My name is Shayan. We're here to discuss a new discovery that could change the future of how we treat Parkinson's disease. Involved in this research, we have Dr. Adekunle Bademosi from the University of Queensland Brain Institute. Dr. Bademosi, it's a pleasure to have you here today. Thank you for joining us. I'd love to have you tell us a little bit about your background.
Okay. Hello, Cheyenne. As has been introduced, my name is Daniel. I'm a neuroscientist at the moment, but my journey to research started many years ago. Like many people, I have a personal experience. My grandma, I remember back in 2010, 11, while I was just coming out of my undergrad studies, was diagnosed with dementia, and I guess it was a bit of a bad time for the whole family because she was a person who had a photographic memory, so you could imagine the sharp decline from remembering every single thing, being able to speak like 5 or 6 languages, to totally going down to a place where she doesn't even recognize any of her children or grandchildren. So that was one of the things that informed my decision to go ahead and have a explore the whole field of neuroscience. I'd already studied medical physiology, so I was very much familiar with neuroscience. And so I moved over to Australia from my country of birth, Nigeria, back in 2013, and I joined a very great lab where I did my master's and my PhD in neuroscience. Trying to understand how the brain functions. I made very interesting discoveries. And then back in 2018, I moved over to Belgium, and that's when a good part of the work we're going to be discussing on today was done in the lab of Professor Patrick Verstrucken. So I was a postdoc doctoral fellow over there, and we're curious as to what happens in Parkinson's disease, and I guess we'll talk in more details about it. But yeah, for me, the passion for my research is not just as that of a scientist, but also as someone who's actually lived and actually taken care of someone who lived, with a neurodegenerative disease.
So Daniel, before we get started, there's many of us that don't know really what Parkinson's is. Do you mind maybe just very briefly telling us a little bit about what that disease is?
Okay, thanks, Dan. That's a very good question. So Parkinson's, as I guess when the name of that disease is mentioned, for at least growing up for me, many people would immediately think of Muhammad Ali, because he was one of the most famous people that developed Parkinson's. But Parkinson's is a brain disease that essentially affects a specific part of the brain. So the brain controls many things that we do, and part of the things it controls is movement. To be able to pick up your pen, it's a fine movement for the brain to tell your whole body to initiate the movement and stop the movement. In Parkinson's, what happens is that the center of the brain that controls the fine body movements becomes destroyed. So you have an overall change in posture, you have a slow movement, you have a bit of rigidity, you also have a shaking phenotype where people are not able to hold their hands steady. It's overall regarded as a neurodegenerative disease that ultimately leads to a change of the body's motor functions. And it's a debilitating disease that's within 7 to 15 years after diagnosis, most people lose their lives to Parkinson's.
With Parkinson's disease, I know I want to get into your research in a bit, but can you tell us a little bit
Yeah.
about what causes it? Is it genetic, environmental? How does it occur?
That's also a great question. And like many other brain diseases, we know that indeed there's the hereditary part of it. But at the same time, there's this other whole part which we don't know, so sort of idiopathic that comes out of nowhere. And in terms of numbers, with Parkinson's, about 10% or less is based on a genetic change or genetic mutation that is passed from parents to offspring. And you have that, being very well studied. You have multiple genes that have been studied, like alpha-synuclein would be a big one in the field. You have LAC2 and many other genes that have been studied where mutations from one or both parents leads to the children or offspring having Parkinson's. But then in the majority of the other cases, it comes out of nowhere. But we know that of this sporadic group of Parkinson's, that's a good number of them could be due to environmental factors. for example, exposure to certain types of pesticides or drugs could induce Parkinson's. And also, you also have, apart from environmental risk factors, you also have even genetic risk factors, which are not exactly the same as mutations passed from parents to offsprings. But it just happens that some people have their gene looking a bit different from other people, and that increases the risk for Parkinson's disease. In the majority of cases, we don't know what causes Parkinson's disease. It just suddenly happens, and the best we've been able to identify are some of the associated risk factors, like I mentioned earlier, that lead to Parkinson's disease.
Now, kind of going into your research, um, I want to touch on it in terms of the discovery, how your team
Sure.
made a major discovery. But before we
Yeah.
get into that discovery, can you tell us what prompted your team towards that direction of research or study?
Okay. That's great. Talking of my team, I'd like to acknowledge the people who were very vital in getting this work out. As I mentioned, the work was done in the lab of Professor Patrick Verstrucken over at Belgium, a majority of it, and then I also work closely with Mariana Desert and Sabine Kernan. So those are key people. As I mentioned, this research, I thought to acknowledge them. Now, what drove us to Parkinson's research? I guess this is work that is built on other previous work we'd actually done in the lab, where as a lab we've always looked at the brain, looked at how the neurons, which are the key components of the brain, how they communicate with one another. But more interestingly, we've also been looking at the disease, Parkinson's disease, and we've sort of studied, as I mentioned earlier, many of the genes that are involved in inducing hereditary forms of Parkinson's disease. So we had made one or two observations earlier where we were studying this process in the brain, which I would introduce now, called autophagy. And just to simplify, what's autophagy? Autophagy is, it's, if you were to think of it, it's a waste disposal slash recycling machinery that is present in every single cell of your body, or almost every single cell. So imagine the brain having each individual basic components, which are the neurons, and every single neuron has a waste disposal machinery. So we had been studying that, and we had shown previously that in Parkinson's, this waste disposal machinery is compromised. And you can imagine, imagine what happens if you keep, releasing waste in your house, and then the waste ultimately builds up, and you don't have any government facilities or anybody coming in to dispose of your waste. You can imagine how dirty your house would be. That's what happens as well in every single neuron, that the neurons continuously generate debris, and so every neuron needs its own waste disposal machinery or waste recycling machinery in place. And that's what led us to Parkinson's, because we saw that there was a link between this waste disposal machinery as well as Parkinson's disease.
Can you give us go a little bit deeper into just the overall functionality of the research and study, and what conclusions you guys drew from that research?
Okay. Okay, this is a very broad question, which probably take me a while to answer, because I need to sort of first give a background as to what led us into the start of the research. Now, one of the things we had discovered earlier was while we're studying some of the well-known genes linked to Parkinson's disease, as I mentioned earlier, LAC2, alpha-synuclein, PACin, was that many of these genes were sort of coming together, and they all had one gene they were all having an indirect or direct interaction with. So it's the best illustration I always give is, imagine you go to different cities in Europe and you keep finding out that all the roads are leading in one direction, as an old saying says, every road in Europe leads to Rome. So in the same way, we discovered that, we looked at PACin, but PACin has had an interaction with this gene we're interested in, Endophilin. We looked at LAC2, it was also interacting with Endophilin. We looked at oxalate and many other different genes, all ultimately had their own interaction either at a protein level with this gene called Endophilin, which is what our research was about. So we're curious because we're like, hey, but there's no genetic mutation in Endophilin. So why is it all linked? Why are they all linked to in Parkinson's disease? Why are they all the different genes linked to endophyllin? And this is the start of the whole research, to sort of find out this seemingly something that was totally in our mind unrelated to Parkinson's disease. And I'll stop here for now and just say, as we started the study, one of the things that actually happened was that we discovered a different study where They're done a sort of a genome-wide associated study. So they imagine taking 10,000 people or 20,000 people in the population and sort of characterizing their genome. And interestingly, here comes the same one we're interested in, endophilin. Endophilin was discovered as one of the genes that was at a high risk for Parkinson's disease, and that even piqued our interest more to say, okay, it may not have a genetic mutation directly linked to Parkinson's disease, but people that have a variance in this gene have a very increased high risk for Parkinson's disease. So that was the start of the research. Yeah.
So when they're high risk, what
Yeah.
contributes to that factor of that Parkinson's gene being activated?
Exactly. Very good question. And that's what our own research sort of ended up finding out because at the start, nobody knew. In fact, in the field, we have loads of studies identifying variances in genes linked to neurodegenerative diseases, but nobody knows the mechanism. We just identify them. And, I'll say our study is one of the first, if not the first, to actually link a variance in a gene with an actual neurodegenerative disease by providing its mechanism of action. So what did we observe? One of the things we had also observed, I'll take a step back earlier, is that we observed that for autophagy to be induced in the body, what normally happens is that you need to have a bit of amino acid deprivation. So, for example, if you decide to go fasting at the moment, you deprive yourself of essential nutrients or proteins, and all the cells in your body sort of get the message, okay, the body is not receiving these nutrients. It's time to ensure that the debris that are being produced by the cells are very well cleared. One of the things we, however, observed during the process of studying that was that we observed that it was not just amino acid deprivation that leads to autophagy induction, but we also discovered that every time the neuron sort of talks to another neuron, so neuronal communication, when you increase the rate of the brain talking to itself, in other words, we also observed that you also had this increase in autophagy. And so this is where our study started from, because in our previous work, we had shown that the protein endophyllin was crucial for autophagy to take place. And so I'd love to simplify this a bit. We knew autophagy was involved, the waste clearing machinery was involved in Parkinson's. We knew that for this to happen, you needed to deprive the cells or the neurons of amino acids. But the mechanism by which decrease or loss of amino acid leads to autophagy getting started or starting of this whole factory of waste disposal we had previously shown was through this gene or through this protein, endophyllin. So our new study was then to find out, is this endophyllin risk factor for Parkinson's disease? Is it in any way linked to neuronal communication? Because that's something we didn't know. So that's what we went on to find out, to see what's the link between the nerves in the brain talking to each other and endophyllin, whether it's a normal one or as a risk factor for Parkinson's disease. What's the link between both? And that's what we ended up finding out in our study.
What type of impact does that have um, compared to other genes that have a role within Parkinson's disease today?
Okay. So that's a good question to ask. So one of the things we always try to find out is when there's a mutation in a gene, for example, how is it inducing Parkinson's disease? How is it ultimately leading down to the dopaminergic neurons? And here I'll just quickly take a step back as well, that what we know is that the part of the brain that is affected whenever people have Parkinson's disease is this part called the substantia nigra, and it is made up of this type of neurons that release dopamine. So ultimately, we're always interested in understanding why these neurons, these dopaminergic neurons, sort of are lost. And different studies have been done. Some have linked this to different organelles or different activities that take place in the neurons. Some have linked it to, my use of scientific words here, exocytosis and endocytosis, so the key forms of neuronal communication. Some have linked it to the powerhouse of every cell, and that's the mitochondria. So you have genes, for example, that are actively involved in the energy generating part of the cells called the mitochondria. In our case, we had previously shown that endophyllin is linked to autophagy. So what we therefore then decided to do was to find out exactly what's happened to this risk factor in endophyllin that increases the risk for Parkinson's disease. And so as compared to other people, we pushed the field further because one of the things we did was to find out the mechanism by which something that's not a genetic mutation, that is just a variant, in people, we found out the mechanism by which it leads to Parkinson's disease. And that is through ultimately changing this process called autophagy in the brain. For the first time, we provide a mechanism by which something that is a risk factor, earlier we mentioned as an environmental risk factor, exposure to pesticides. Here we've taken a genetic risk factor and we've provided a mechanism by which a genetic risk factor leads to Parkinson's disease. And maybe I'll just try to buttress a bit more on exactly what's happening here. So at its core, what happens when Parkinson's disease takes place is that these neurons that produces dopamine, they're lost. So you have a loss of dopaminergic neurons, which is why for the treatment, I will get to this, probably talk about this later. Many of the therapies available just try to supplement the person living with Parkinson's disease with dopamine, because by supplementing with dopamine, you have the chemicals that are doing what the dopaminergic neurons should be doing. Now, what causes the loss of the neurons is something we never understood. We just know they're lost. Here, with this study, we're able to start identifying what causes the loss. Why? Because what happens when autophagy, this waste disposal machinery, what happens when it doesn't work properly is that, as I mentioned earlier, you have a buildup of debris. So imagine a precious neuron in the brain where you just have, this debris build up. It keeps getting built up over 1 year, 2 years, 5 years, 10 years. After a while, it's just like when you go to a dump, the smell it produces, the waste, everybody wants to stay away from a dump. The same thing happens in the neuron. The debris become toxic, and that toxicity leads to the death of those neurons. So at its core, addressing autophagy here is also a novelty because we find that, okay, this risk factor actually alters the way every neuron in the brain deals with the debris that ultimately will kill the neuron if it's not well dealt with.
Interesting. Now, in terms of that process that's going on, are
Yeah.
there other processes involved within Parkinson's that also contribute to it, or would this be the main functionality that's occurring that's causing the neurodegeneration within Parkinson's?
So there are many other processes. It's one of the beauties of the brain that the brain, multiple things are happening. There's inflammation aspect where you have the neurons or sites of the brain getting inflamed and you have the inflammatory responses. There's the loss of energy as produced by this organelle I mentioned, or this part of the cell I mentioned called the mitochondria. So there's also the parts of the neurons not being able to communicate with one another. So these dopaminergic neurons are meant to release dopamine. So what happens if everything else is working properly, but they're not able to release dopamine? There's also a buildup, and that also leads to disease. So multiple mechanisms have been highlighted as being involved in the onset of Parkinson's. I guess here with what our study, we just show a novel mechanism by which this waste disposal clearance mechanism is altered in Parkinson's. And the advantage of that is that it's actually druggable. So it's a druggable target where we can actually say, okay, what happens if we improve the efficiency of this machinery, or what happens if we decrease the efficiency of this machinery in someone living with Parkinson's disease.
What type of impact does that particular process have compared to the other components involved within Parkinson's?
Importance, I would say it's very vital. It's the core. In many ways, I see the autophagy machinery as, if I were to compare it to us having a car and driving a car, it's essentially the mechanic that's does the servicing of your car. But there's more to driving the car than having a mechanic. But ultimately, you can drive your car for, let's say, after buying it brand new, you can drive it maybe for 3 years or 4 years without going to service it or taking it for any repairs. But then after a while, you definitely pay the price. In the same way, many other things are happening that leads to Parkinson's disease, but I see autophagy as being very crucial and vital. So the neuron is able to function for a while without it, but when the consequences sort of catch up to the neuron, then it becomes very bad. Because its relevance is that it ensures that the debris in the neuron is cleared off. And this is something I'd just like to touch on, that, for the brain, as with every other part of our body, we're always generating wastes. There's a reason why if you lock yourself up in your house for 1 week, you're still going to find dust in the house because your skin cells, are continuously being shed, and then you have there's a continuous renewing where new skin cells are produced. The same thing happens in every single part of the body except one. So every part of the body continues to, even the heart has new muscles being produced. But in the brain, once the neurons are produced and they mature, that's it. There is no other production of any extra neurons. And so the mechanism by which the brain sort of deals with renewing itself is rather than generating a total new component, a new neuron, a new dopaminergic neuron, what it does is it services it, it renews it by taking some parts that have gotten damaged, disposing them, or actually recycling them. So imagine if this brain's mechanism of disposing or recycling waste that is naturally produced is damaged, then of course that leads to trouble, and that's what we're showing in our study, that with this risk variance just targets that waste disposal machinery. So in a way, the best illustration I can show is it's imagine for a suburb you have a factory that disposes of the waste and normally should function at 100% efficiency, but something happens and it can only function at 40% efficiency. So things are still working, but then they're not able to, for example, go around all the streets and clear off all the waste from every single street, every single neighborhood. Ultimately, it will show in the way the neighborhood looks like. In the same way, this risk factor targets the waste disposal in the neuron, and that ultimately leads to, buildup of debris
Okay.
in the neurons.
There's a lot going on. Do you mind maybe, I mean, for individuals that are watching this, um, can you maybe just sum up ultimately what you guys concluded from this research and what type of impact that's going to have?
Okay. So our conclusion in this is now to get into a bit of more fine detail as to exactly what's happening. One was we made a discovery on the function of endophyllin in general. And what we discovered was that every time neurons talk, we knew all along that when neurons talk, they use sort of electrical signals to talk to each other. And the electrical signals change to chemical signals at certain points, which are called synapses. For the electrical signal to change to a chemical signal in the brain, you need this metal called calcium. So we have what you call an influx of calcium into the neurons. What we showed was that every time the electrical signal was changing into the chemical signal, this influx of calcium, which we thought was doing many other things, we actually discovered for the first time that this calcium influx actually goes on to touch endophyllin. Indirectly, we don't know the specific mechanism, but when the calcium that has gotten into the neuron touches endophyllin, there's a change in its shape. And that change in its shape is what helps it say, okay, it's time for me to induce the production, or it's time for me to kickstart autophagy, or this whole waste disposal machinery. And so in our study, we did that to characterize this very clearly and show that for autophagy to take place when neurons are communicating, the change from electrical to chemical, which leads to calcium influx, induces autophagy by telling endophyllin to act. However, with the risk factor variance, when it's present within a neuron, you still have your chemical signals, you still have the electrical signals coming down the neurons, but the difference is that when calcium influx gets into the neurons, the protein is totally non-responsive to the influx of calcium. And so what we identified was not only that the risk factor dampened autophagy, we showed how it's dampened autophagy by being non-responsive to the influx of calcium. So influx of calcium does thousands of other things in the neurons, like too many other things. But we showed this, which is the second part of the novelty of our study. We show that the influx comes in. Yes, you have all the other processes going on, but this essential process, which should kickstart autophagy, is just not working because the variant makes it non-responsive to the influx of calcium. And so to summarize all of that, we essentially showed two things: a mechanism of action by which a risk variant leads to Parkinson's disease, and then we now show exactly how it does it, like at its fine details, at its molecular level. And that informs of our next studies, how to then start targeting the process that is affected itself, which is called autophagy.
So with autophagy,
Yes.
Are you able to connect that with any other neurodegenerative diseases, or is this focused primarily on Parkinson's?
Very, very good question. That's an excellent question because, and this is the beauty of our study. Almost all neurodegenerative diseases are linked with autophagy, and that's what has been clearly shown over the past, I'd say, 10 to 15 years. The mechanism by which they're linked is not known, but what we know is that when we take the brains of people that are living with that have lived with neurodegenerative diseases, you have this loss of different parts of the brain. You know, as you mentioned earlier today, there's atrophy. Now, what causes the atrophy? What causes the loss? In many ways, it's linked to those neurons dying. And what we've discovered is that autophagy plays a role, a massive role in each of them. So in Alzheimer's disease, you have multiple researchers studying autophagy, same in Parkinson's, vascular dementia, frontotemporal dementia, motor neuron disease, anything that has to do with a neurological disease. Autophagy is many a times strongly involved in the buildup of debris. And this is the key part that if you've heard of Alzheimer's disease, you'd have heard words like tau pathology or A beta accumulation. And those are debris, you know, that needs to be cleared but not cleared. In Parkinson's disease, the key word people would use in the medical field is the buildup of Lewy bodies. The Lewy bodies are those same debris that have built up over the years and have become, you know, so almost this aggregates in the brain. So the reason you have those debris and aggregates in the different neurological diseases is ultimately because when those toxic materials are produced by the neurons, autophagy, which should be clearing them, is not working efficiently, and that ultimately leads to a buildup. So our discovery now not only has an impact on Parkinson's disease, it has an impact on all neurological or brain diseases.
That's amazing. Can you, with
Yeah.
autophagy, is there any way to link it with with particular maybe symptoms or particular symptoms that maybe people experience in terms of neurodegenerative diseases? Are we able
Yes.
to go that deep?
To the best of our knowledge, I'll say we don't have a particular correlation between autophagy and a symptom. However, I'll mention this. Um, as I illustrated earlier, you could buy a car and drive the car for a while before you take it for servicing because it can function for a while. However, if you don't service it, things start, you know, you start to have gradual build-up of different things not working until one day the car stops working. In the same way with autophagy, we are of the opinion that it actually explains why with many neurological or brain diseases, there's sort of a progression before you actually have the key symptoms. And to explain this further, I'll pick Parkinson's as an example. When I mentioned explain what Parkinson's is earlier, I was talking about rigidity, you know, tremors, and not being able to move properly. But what we know as researchers and as medical personnel in the field is that 10 to 20 years before you have any motor symptoms or movement symptoms, people that end up with Parkinson's have loss of smell or change of smell. So you give them a banana and then they tell you they're smelling an engine oil. They have sleep fragmentation. They're not able to sleep properly. Their digestive system totally changes, you know. So this is subtle things that build up about 20 years, which we call the non-motor symptoms of Parkinson's disease, sort of serve as indicators to announce that something is already happening in the brain, which is not functioning efficiently. So when things have become very bad, 100% the machinery is not working anymore, then you start to have the motor symptoms. And so to the best of my knowledge, the way to sort of correlate it is that when autophagy is not working properly, you start to, the neurons are unable to work efficiently, but they can still work. And so you start to have these early signs and warning signs letting you know that something is not working properly. In the case of Parkinson's disease, it will be the loss of smell, it will be the sleep fragmentation, and some of the others I mentioned.
What I'm curious is what needs to be done in terms of a therapeutic approach. So we have this discovery. What's next for a
Yeah,
therapeutic approach to address this process?
that, that's a question I've actually kept asking myself. And in answering this, I remember I got an email earlier in the week, and someone was saying, "Oh, you're talking about potential compounds that can influence autophagy, but if you increase the level of autophagy, or if you decrease the level of autophagy, won't that also be harmful?" A way for me to explain this is to say this machinery needs to work at a very optimal rate. If it goes into an overdrive, it's harmful. If it doesn't work at its maximal efficiency, it's also harmful. So therapeutically, what we need to do is we're looking for ways to go in either direction, to either increase autophagy when it's not efficient, or to dampen autophagy when it goes into an overdrive. Now, why do I mention this? One of the most popular mutations that leads to Parkinson's is in a gene called LRRK2. So that's LRRK2. So LRRK2. Now it's called as G2019S. For people that have this mutation, there are not that many in the world, but they're a significant number. They form one of the large majority of people that have genetic forms of Parkinson's. But for people that have this specific mutation, in their brain, what's happening is not that there's a buildup of debris. The opposite is true. For them, what's happening is that autophagy goes into an overdrive. So imagine a waste disposal machinery going into an overdrive and not only disposing your waste, but going in and getting into your house and taking your sofas and every other thing and, just as dismantling all the houses on the street.
Right.
That's what's happening in these groups of people. And so there's therefore then that fine balance for autophagy where in some scenarios, very rare, but it happens, it goes into an overdrive. And in the majority of scenarios, it's not working properly. And then you ultimately, therefore, then still have a buildup of debris. So what we therefore then have to do is to find the compounds, and those compounds are already available, and it's many of them have been done used as clinical trials for people that live with cancer, because cancer and autophagy is also strongly correlated as well. So we have drug compounds that could actually enhance autophagy. There are many of them out there, and then we also have compounds that could actually inhibit autophagy. The question is, have we gotten to a stage where we should start them on a clinical trial, or even if it's to move them onto other animal models to test them and see if they improve survival, for example, um, in the case of people living with Parkinson's. And I would also like to mention here that one of the strengths of this study where this study would shine out a lot, especially in using these compounds, is if we're able to find a early means of diagnosing Parkinson's. So imagine someone 20 years before the movement symptoms suddenly discovers that they've had a change of smell and decide to go for a test, and they discover that, oh, you have a high risk for Parkinson's disease. And from then, they start to deal with this waste disposal machinery, then you can imagine that 40 years down the line, they'll still be okay because the debris is being taken care of before any loss of neurons. One thing I always say is that once you lose the neurons, once you lose that part of the brain, how do you gain it back? There's virtually almost impossible to gain those same neurons that you've lost back. You can gain new neurons, maybe with stem cell technology if we get there one day, but the very one you've lost is gone. So the strength of our study would shine when we have a means of diagnosing some of these neurodegenerative diseases very early, and then hopefully taking some of these compounds that will either inhibit or enhance autophagy and applying them at the very early stages when these non-motor symptoms have been observed.
So how are they able to go and diagnose at an early stage? Like what needs to be done to be able to identify so early?
Yeah, so I would here acknowledge many of the other researchers who have massively involved in this field. I know there are people who are looking at biomarkers, so where they can take samples either from your spinal fluid or from the blood, and then we're studying to see if there's any compound whose levels increase as a sign that a person might be developing some of the neurodegenerative diseases. So this is a field that lots of people are also working on to help in early diagnosis. And we'll wait and see what happens in that field to see if we come up with something tenable, which is actually a marker that can be tested, to say, for example, if someone has a bit of um, infection, all you need to do is to check for the white blood cells, and you know that the white blood cell levels would increase because the body is dealing with infection. In the same way, my hope is that in our field we're able to get a biomarker that we can use to actually identify yeah, the onset of Parkinson's disease many years before.
Okay. Going back to autophagy, I know you said there it can either um, be there could be it could be induced or enhanced, right? In terms of the
Yeah,
process, which
yeah.
how would you know which direction it needs to go?
Oh, I'd leave that to the medical doctors to be able to tell us with direction. But all I can say is, to the best of my knowledge, and I'm not, I don't have the full knowledge here, but to the best of my
Yeah.
knowledge, the scenario of autophagy being going into an overdrive only happens in a very few people that have the genetic mutation. So
Okay.
I don't know if any of the other risk factors, like environmental risk factors, also causes an increase in autophagy. But then in the majority of cases, what we have is rather the dampening of autophagy, which I'd say therefore then informs us to focus more on compounds that actually would enhance autophagy if what happens in the majority of cases is that autophagy is dampened within the brain.
Okay.
Yeah.
Can you tell us what are some of those compounds that could be used based on this new discovery?
Oh, um, there are quite a number of them. Like even if you search autophagy enhancing drugs, there are quite a number of them that have already been noted. And the reason I hesitate a bit in saying this, is that I don't want anybody to hear that and say, "Oh, I'll go take this compound and see if
Right.
I
Right.
improve," because I'm not a medical doctor, so I won't. But I can definitely say that there are quite a number of probably 50 or more autophagy enhancing drugs that are out there that can indeed be used for for the purposes of research at the moment. Now, for the purposes of um, medical trials, I know, for example, the drug rapamycin is currently in clinical trial for some of the other neurodegenerative diseases. So you have some other neurodegenerative diseases that are currently being where autophagy enhancing drugs are currently being tested. So that's we're waiting to see what happens with those clinical trials to see if they actually improve survival, and if they do, that also opens up the field for people to then test them in multiple other neurodegenerative diseases.
How soon do you think some sort of treatment could be available and accessible to people if it's shown that it could be effective?
I'll assume it depends. So one of the things I guess that tends to be a setback to some of the use of these compounds is the fact that their function is not may not only be limited to the part of the brain you're interested in. So take Parkinson's. Parkinson's doesn't affect the whole brain; it affects the dopaminergic neurons. But then, if you take a compound that improves survival of the dopaminergic neurons, but it's harmful to other parts of the brain, that becomes, then we need to go back and say, okay, now we have a compound that is useful, but not useful to the whole body. I mean, in terms of timing, I guess I have no idea again because this is not my expertise to be able to give an answer in terms of timing. I'll simply say it's a waiting game. We have to wait and see.
Any kind of finishing thoughts or anything else that stood out to you throughout your research or any kind of direction you see all of this potentially going?
Well, I guess a couple of things I could say in that direction. Some I've already mentioned in terms of, the autophagy enhancing or inhibiting compounds, and that being the next phase or direction to focus on. But as a researcher, I think one of the things that intrigues me is the fact that, you know, the attitude of never giving up, but always being curious, always leads to an interesting discovery because um, we knew we'd studied endorphin for many years, like people had studied it in the context of other neuronal function for many years, but to find out that it's involved in autophagy was a new discovery for us. That was a while back. Now to now find out that, the neurons communicating with each other signals endorphin to induce autophagy is another new discovery. And for me, it just tells me that there's more to discover. And at times people ask, why are you guys always focusing on, the new discoveries within the brain? Why not on more of the chemical compounds? I use this scenario, we wouldn't have thought about enhancing or inhibiting autophagy if we had not understood this novel, at least to us, a novel function of endorphin responding to increased neuron to neuron communication. And so that curiosity for me is something I'm taking away from this, which I'll be applying going forward as well, to always be curious to understand, even if it seems impossible to keep trying to understand what's actually happening in the brain in health and then in disease. Now, therapeutically, fingers crossed, we'll see how things pan out as we take more of these autophagy enhancing or inhibiting drugs and then we apply them, especially now in the neurodegenerative field. I know, as I mentioned earlier, that the field of cancer research has applied them a lot in their clinical trials. So it's going to be exciting to see what we get as we continue in our field, and hopefully we get some good news in a few years to come.
That's some amazing thoughts and just observations you've made, Daniel.
Oh, good.
Where where do you see your research going next?
That's a good question. So personally, my research, I've expanded beyond Parkinson's disease. I'm also looking at frontotemporal dementia, which I guess if it wasn't popular before, Bruce Willis has made the name popular now because it's the disease was diagnosed with. So I started researching on this about 2 years back, and I'm also working on motor neuron disease, also looking at aspects of autophagy within this aspect as well. As I told you earlier, autophagy is implicated in multiple other brain-related diseases. I would like to mention this, even though I'm not actively involved in this, but then it's just something that I always found curious. Um, evidence shows that many years ago we had the Spanish flu. So I'm going into the COVID research on Parkinson's disease because there are papers out there on this. But evidence shows that many years ago, I think just after World War One, we had the Spanish flu. Now, many of the people that ended up being diagnosed with the Spanish flu within 40 to 50 years after that, there were a good portion of them, the correlation is very high, were diagnosed with Parkinson's disease. So there's a strong correlation between people that were exposed to Spanish flu and people that ended up with Parkinson's disease, and that started this whole field of viral infection leading to neurodegenerative disease, which to this day you still have many researchers doubting the data, saying is that possible and all of that. And having said that, when with the start of COVID, soon as it started, having already been in the field of Parkinson's disease, I was curious to see what will happen. The reason being, COVID was showing many of the symptoms you have 20 to 30 years before you have Parkinson's disease, the loss or change of smell, sleep fragmentation, and all the other things, especially the smell one. And so I know that there are a lot of researchers who are now focusing on potential effect of COVID in the long term, beyond just recovering from it. I've read a couple of papers that came out from Oxford, for example, showing that people that had COVID had a higher rate of brain shrinking than normal, so which was, interesting. I know researchers who are studying to see if there's a buildup of COVID viruses in the brain even after recovery, which happens with some diseases. I know tuberculosis, you could have a buildup, for example, of of bacteria, and after a while, people who previously recovered from tuberculosis suddenly have it again. Those are for me interesting lines of thoughts. I'm not studying them actively, but I'm curious to see what's going to come out of the field. Especially for Parkinson's to see and COVID to see if they will have a similar correlation in a few years' time between people that were exposed to COVID now, as well as increased risk for Parkinson's disease. I guess it's a matter of time, but we'll wait and see.
That's awesome. Do you know when information like that would be available?
I guess it's the same way we waited for the Spanish flu. People, it would be a waiting game, but I know that rather than wait, that there are loads of people who are looking at uptake of COVID, of the SARS-CoV-2 viruses into the brain to see if it's retained there or not. They've used many mouse model, and some are reporting yes, some are reporting no. So I guess it's a waiting game as well there to see because it's the whole world population is where we're getting the data from. So we'll wait
Right.
and see what happens there. But for me, my curiosity was more based on the change of smell, which was the link between COVID-19 as well as Parkinson's disease.
Would love to. I mean, when that information's out, it's definitely something I'd want to go and look into.
Mm.
In terms of your research with just the other different neurodegenerative diseases, which direction are you going with those?
Um, I'm at the same stage I was when I started the Parkinson's disease research, starting with trying to understand what's happening in disease. That's where I'm starting from. Also, I have a risk variance, just like I had for Parkinson's disease. I also have for frontotemporal dementia and motor neuron. So I'm studying that as well, hopefully to find out a new mechanism as well. I also have a compound, which I can mention now, which I'm also studying to see if it would alleviate or help, with recovery. That's where I am. But at the moment, I think my whole field is about, at least for me, my interest is in looking at this risk variance because
Yeah.
many people who end up with the forms of neurodegenerative diseases that we don't know, a good number of them simply have a genetic variance and not a mutation. And so if we could find therapies for that, you can imagine that a large portion of people living with neurodegenerative diseases will be very happy.
All right, especially with with that form of dementia. I mean, there's really no treatments out there. So being able to do something like that would be life changing. Um,
Big time, like
do you
big
know, I
time.
mean, what, tell me a little bit about the timeline for that type of study. I mean, I know it's something hard to predict, but um,
Yeah.
what kind of timeline do you see this whole study kind of occurring over?
Well, it's I think you get timelines when you're studying you get timelines when you have a clinical drug and you're going on clinical trials. In our research, we don't have timelines because we first have to understand the disease, understand
Yeah.
it very well before we can indeed find out, okay, what compounds can be used to target, the disease. So for example, Parkinson's disease was discovered or it was named was in 1908, or as far back as then, and we're still studying it today,
Mhm.
without having. We only have one drug called levodopa as treatment, which is not even a cure, but rather a compound that manages, the symptoms. So all I can say is the more we've understood the neurodegenerative diseases, the better we've been able to improve survival or even or managing of symptoms. And I know in our time there's a huge drive towards finding a cure or towards diagnosing early, because if you diagnose early then and you can start applying the drugs to help maintain the symptoms or prevent the symptoms, then you can improve, survival. So back to the question in terms of the time. I guess there's no time. We just have to keep researching using as many technologies. One of the technologies I use is super resolution microscopy, which allows me to see some of the proteins that we're studying in details. I think the resolution is about 10 million times that of your standard iPhone. So it provides this huge resolution for me to do my study, and I'm applying multiple other novel technologies as well, with the hope that we can actually understand the disease precisely and clearly.
That's very exciting.
Yeah.
Daniel, thank you so much. Really appreciate your time.
Oh,
Thank you
I'm
for walking us through all of this. I'm sure everyone that's watching this, they learned something new, and it's very exciting stuff.
good.
So thank you for what you're doing, and would love to go ahead and
Oh, good.
reconnect soon. As soon as you progress through that research, I'm definitely very curious
Definitely.
myself.
Definitely. Thank you, Sharon.
Thank you.